Counting down to the exam. Four more big days. You find that funny? I have not yet scheduled a review session, but it is almost certainly going to be, unfortunately, on Thursday night. I don't have any other time free when I can do it. So I will announce that later. It will probably be in ALS 4001, and it will probably be around 5:15 or so, depending upon if I can get the room. So I will let you know about that. But I don't like to do it the night before the exam, but that's about the only time I can do it this week. All right. The other thing is, I said last time the material would go through Wednesday. You'll notice on the sheet that Wednesday is a review session. And so the material is actually going to go through the end of this DNA replication, repair, recombination, which might be today, depending on how we do. If we get through it today, then Wednesday we'll actually have an in-class review. If I don't, then I'll still finish a little bit of that up on Wednesday. Yes, Julie?
Are you going to post the video to the review session Thursday night?
I will, yeah. The question is, will I post a video for the review Thursday night? Yes, I will, as soon as I can get it posted. I'm dependent upon OSU's rendering of it, and so sometimes it varies. Yeah. So, but I will definitely strive to get it posted as soon as I possibly can. Yeah. Yeah. Okay. Other questions? Okay. Well, we've made it. Does he have? Yeah. Am I going to post a practice exam? No, I don't post practice exams after the first one because the first one is to show you what the format is, and so you guys know what the format is now. So no, I won't. Okay. Last time I pretty much got through what I wanted to say about replication in general. Today I'm going to talk about some specifics about replication. Then I'll talk about repair. And at the very end of the lecture, if I haven't finished everything else, I will talk a bit about recombination. One of the things that I sort of alluded to last time was the fact that the question was, well, how does the polymerase know where to start, et cetera, et cetera? And it turns out that there is actually a complex that is built to start replication within any chromosome. Okay, so in our chromosomes, it turns out we have many hundreds or thousands of places where replication starts. And our replication is, in many ways, the same and in some ways different from that of bacteria. So I'm going to point out some of those differences later. First, I want to tell you something about bacterial replication because it's the most studied and the most understood. Bacterial replication occurs, of course, on a circular chromosome. I want to emphasize that. And it's because of the circular nature of that chromosome that there are fundamental differences in the, at least the completion of the replication process, as we shall see. Well, every place where replication starts is a specific sequence. It's called an origin. Okay? So DNAs have built into them sequences that are recognized by proteins that recognize origins. That's not totally surprising. And those proteins that recognize origins help the DNA polymerase to get started. And once that replication fork has been assembled, the polymerases can go off and do their thing. E. coli has in its circular chromosome, the circular E. coli chromosome is about 6.3 million base pairs. That 6.3 million base pairs is replicated starting from a single origin. A single origin, okay? That origin has a name. It's called ORIC, O-R-I-C. Now, it's at that origin where this process begins and the things that I will be describing to you actually occur. At ORIC, this replication origin is what's called bidirectional, meaning that one replication fork starts and goes one direction, a second replication fork starts and goes the other direction, and they get up and they ultimately meet at a termination sequence. I'm not going to talk about the termination, but you can imagine there has to be some orchestration in terms of getting everything finished up properly at that termination, and there is. Okay? We won't talk about that. But the ORIC is a single bidirectional replication origin. Okay. Now, the ORIC has an interesting sequence structure. There are 4 binding sites for a protein known as DNA A. These have a structure that looks kind of like this. And you will notice that in comparing and looking at the consensus sequence of these, you'll see that they're very AT rich. Okay? This AT richness turns out to be very useful because what the DNA A protein does is it facilitates the wrapping of the DNA around itself. And the wrapping of the DNA around itself causes there to be some stress on the DNA. Like we saw the topoisomerases recognized stress. What happened when we had stress? Well, the strands may adjust themselves accordingly. And that's exactly what we see with this wrapping of the DNA around the DNA A protein. Okay? So the DNA wraps around the DNA protein, there is some stress, and the strands actually come apart. That's facilitated by the fact that there's only AT base pairs holding them together, and you recall that AT base pairs do not have as many hydrogen bonds as GC base pairs, and this facilitates the starting of the process. You'll notice that there are also, let me back up here. So the AT-rich regions are adjacent to the binding sites, okay? So we're creating the tension here, and we are causing this region adjacent to it to unwind. Okay. Now, this is what this complex looks like. You see that there is a hexamer of DNA A protein, and the DNA chromosome wraps around it. This creates this torsional stress. The torsional stress is manifested here. These sequences come apart and now provide entry for the proteins of the replication fork. Okay? So what will happen at this place where the green sequences are is that we will see entry of proteins called DNA B, DNA C, DNA G, and the beta clamp. All right? Now, for our purposes, the most important two here are the DNA B and C. They actually come as a complex, and DNA B-C act as a helicase. DNA B-C is known as the helicase. Specifically, the C is, but that's not important for our purpose. We think of it as the B-C as being the helicase. Well, putting a helicase in there really early makes a lot of sense because what does a helicase do? It starts peeling apart strands even more. And if we're going to assemble a replication fork that's going to include all these other proteins, such as single-strand binding protein, primase, DNA polymerase, and so forth, we want to have these strands far enough apart so that we can fit everything into that place where the replication is going to occur. That happens, and replication proceeds. Now, as I said, it goes all the way around the circular chromosome and terminates at a specific point, or one of two or three specific points, on the other side of the chromosome. All right? So when that's done, the E. coli chromosome has completely replicated, with the exception of the fact that you now have two intertwined circles. You may remember I said there's a topoisomerase that takes those two intertwined circles and does that. Allows them to separate, and by allowing them to separate, the cell can then divide. This table shows that there are many DNA polymerases, and in fact, it doesn't even show you all of the DNA polymerases. In prokaryotic cells, for example, there are actually 5 DNA polymerases. All right? They only show 3, the 3 that have numbers. The other 2 have different names, and they don't always function as DNA polymerases. They're kind of odd enzymes. One of them is called the sloppier copier. Okay? I got one laugh out of that anyway. The sloppier copier, all right? I'll let you guess why they call it the sloppier copier. Why do they call it the sloppier copier? It makes a lot of mistakes. And this DNA polymerase activity appears to be important only when the cell is having damage to its DNA. Now, if you think about it, normally you don't want to have mistakes and so forth that a polymerase makes. But if the cell has suffered damage to its DNA, making a mistake might actually put things back where it was. That's one consideration. And second, mistakes allow the polymerase to not get stuck if there's something, for example, that doesn't look anything like a base on the other side. So some of the damage that can occur to DNA involves what are called adducts. Okay? Adducts are big honking molecules that get covalently attached to bases. And they make great big bulges in the DNA. Right? Cigarette smoke is full of materials that make adducts. Adducts really screw up a regular DNA polymerase. It will stop and not do anything. So having a sloppier copier, I got the hiccups. A sloppier copier makes a lot of sense. Eukaryotic DNA polymerases are not nearly as well studied as the prokaryotic DNA polymerase, and you'll see that among the eukaryotic polymerases you see some specialization of function that you don't see in prokaryote. Remember in the DNA polymerase 1 here we had 3 enzymatic activities in DNA polymerase 1. In the eukaryotes we tend to split those activities up somewhat among the different enzymes. I'm not going to say a lot about individual DNA polymerases in eukaryotes. Well, eukaryotes have other considerations than prokaryotes do. All right? For one, eukaryotes have chromosomes that are, on average, about 1,000 times larger, at least in terms of total DNA content, than E. coli has. Okay? About 1,000 times larger. About 3 billion base pairs compared to 6 million base pairs is about 500 to 1,000-fold difference in size between humans and bacteria. Okay? That means that there's a lot more orchestration that has to happen among among eukaryotic cells for replicating their DNA. A lot more orchestration. It also turns out that eukaryotic cells have to all work together. They all have to pull their oars at the same time. If you do that, you all make progress in a boat. If one is pulling the oar and the other is not, you go in circles. So eukaryotic cells, therefore, have to control their cell division, and they have to respond to outside signals. Now, we talked last term about signaling and how growth factors and so forth can bind to cells and stimulate the process of cell division. Cells in eukaryotes have a cycle that they go through. That cycle includes 4 stages. S for synthetic stage, G2, which is a preparation for cell division, and then a resting phase, G1. Okay? It's at the synthesis phase where DNA is being synthesized. Okay? So I want to say just a few words about eukaryotic DNA synthesis, and the few words relate to a very, very important aspect of eukaryotic DNA replication. Eukaryotic cells have linear chromosomes. Linear chromosomes. And linear chromosomes pose a completely different scenario for replication than circular do. Okay? We thought about the circular, how we could go all the way around, and we would only have to worry about the RNA primers in certain places where the DNA polymerase could extend an existing piece of DNA and get rid of that RNA primer. Okay? Everybody remember that? We don't have that luxury at the ends of eukaryotic chromosomes. Imagine, if you will, that, first of all, a eukaryotic chromosome will have hundreds or thousands of origins within it. All of the internal ones can remove the RNA primers. No problem in removing RNA primers on the internal ones. But the sequence is at the very end of the linear chromosome. That's not possible. Okay? Now, that may not seem like it makes sense at first, but let me show you, using, again, a high-quality artistic rendition, what happens. Here is a representation of a linear eukaryotic chromosome. Okay? I'm only going to focus on the replication at the ends. I'm not going to worry about all the stuff in the middle, because all the stuff in the middle, the RNA primers can get removed. I start replication at the 5' end here, and I start replication at the 5' end over here. We see that the other strand sort of peels off, as we see here. Okay? And suffice it to say that this becomes part of an Okazaki fragment. Okay? So we have the lagging strand synthesis will proceed backwards and backwards, as we see here. This top strand, the lagging strand synthesis can work all the way to the 5' end. So this guy ends up being fine. At the other end, however, when this polymerase goes all the way across, what we see at the end of one round of replication is something that looks like this. We have a linear, we have the red corresponding to the RNA, and the other duplex has the red at the other end here. Okay? So each of these duplexes have one short stretch of RNA that can't be removed. Okay? If we remove it, And it's possible to remove it. I don't mean to say we can't get rid of it. If we remove it, we can't fill it in. Okay? So either way, we're going to lose something. Well, what happens, I'm just going to follow the top one right here. If I follow this guy, and now I start replication of this guy, what's going to happen? Well, I put an RNA piece there. It replicates all the way over to here. And when it's done, I get two pieces. One piece that includes the, and I've unfortunately swapped the strands. This guy here corresponds to replication going that way. And this guy here corresponds to replication going this way. But look, when the DNA polymerase hit that red region, it couldn't copy it because this is RNA. And a DNA polymerase will not copy RNA. The next round of replication will start right here and move that way. And it's going to run into the same problem. What that means is that for a linear chromosome, every single time we start a round of replication, a short stretch at the end of the DNA cannot be replicated, and the chromosome will get shorter and shorter and shorter and shorter. My chromosomes are shorter than anybody else's in here because I'm older than you are. My cells have gone through more cycles of replication, and I've lost more DNA. Therefore, you guys should be smarter than me, right? You got a lot more DNA than I've got. There's no, I hear rumbling, but yeah, we are smarter than he is, right? Okay, so that could be pretty deadly, all right? Because if I start losing genetic material, when I start losing genetic material, I start losing genes, and when I start losing genes, genes are what's keeping me alive, I can have problems, okay? Well, cells are, eukaryotic cells are set up in a very interesting way. At the ends of them, there are special structures called telomeres. And that's the part that's being replicated here. The telomeres include thousands of copies, repeated copies of the same sequence over and over and over and over. It's typically about 7 to 9 base pairs long. People used to refer to them as junk DNA. Among other things, they called junk DNA. Because it didn't code for anything, it must not be important. Well, I like to think of those telomeres as a fuse. Okay? There's a fuse. If you light a fuse, what happens? Well, the fuse burns down, and eventually something blows up. Right? Well, if this fuse burns down, that is, once I have gotten rid of all the duplicated sequences of my telomeres, I'm going to start deleting genes that I need. When the cell runs out of telomeres, the cell dies. Okay? Now, there are suggestions that the length of the telomere may be a factor in longevity, how long people live. There's some very interesting work that's being done with that, for example. Well, how do the telomeres get on there in the first place? The way the telomeres get on there in the first place is by virtue of action of an enzyme known as telomerase. Telomerase makes the telomeres. Now I'm going to show you how it makes it, and then I'm going to say some implications about this for you after that, and then take questions after that. Telomerase is a very interesting enzyme. Okay? It's an enzyme that is a reverse transcriptase. It's a reverse transcriptase. Now you think, oh, reverse transcriptase, it copies RNA, therefore it fixes the thing at the end. That's not how it works. Okay? So telomerase carries a primer of its own. It carries a short circular primer that you can see right here. Okay? This primer includes a sequence, CAA, TCCAA. No, you don't need to know the sequence, but that's an example here of this primer sequence that is being carried by this telomerase. The way telomerase works is the enzyme binds to the region, to a telomeric region at the end of the DNA, okay? And it allows its own DNA polymerase activity to copy the primer that it's carrying. So telomerase both carries a primer and is a DNA polymerase, a reverse transcriptase DNA polymerase. Again, a reverse transcriptase copies RNA and makes DNA. So it's copying its own primer, and the piece that it's copying, look at this, is this. Well, once it's copied this, the telomerase slides down, and it copies it again. So by doing this slide, you make thousands of copies of this. And you're saying, but wait, Kevin, what about this strand? Okay? Well, it turns out that once I've made things way out here, guess what? Primase can now come back in here and go ding, 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 and make all that. Okay? So as a result of this action, telomerase facilitates the lengthening of telomeres at the linear ends of eukaryotic chromosomes. Okay? Now, don't confuse this RNA with the RNA that I showed you in the last figure. That's necessary for starting that replication process. This RNA is a primer carried by telomerase, and this RNA provides something to copy. There's the bases that it copies. The reason you get thousands of repeats of the same thing is because this guy only has a short stretch that's copied. Okay, now, well, telomerase is great then, so why do our telomeres shorten? All right, if telomerase can lengthen our telomeres, why do telomeres shorten? The answer is that most cells in our body do not have active telomerase. Most cells of our body, in fact, probably 99% of the cells in our body, do not have active telomerase. All right? That means that most cells of our body, every time they replicate, they get shorter and shorter and shorter. Well, what if we were to make telomerase active in all of our cells? Would we live forever? I don't have an answer to that. There is some recent results that suggest that activating telomerase may, in fact, help increase longevity. But there's concern about doing this sort of thing because the other cells in which telomerase is very active are cancer cells. Cancer cells need to have telomerase active because they can divide uncontrollably and they can divide forever. If you take cancer cells out of a person, you can grow them on a petri dish forever because they keep replacing their telomeres. You take regular cells out of a person, you put them on a petri dish, they'll divide for a few generations, and they will die because they will burn out their telomeres. Well, when do we get our telomeres? We get our telomeres during fetal development. Okay? During fetal development. That's when we gain our telomeres. Stephanie.
Are they allosterically activated?
Are they allosterically activated? No. The telomerase itself is just not synthesized in our cells. Good question. Yes?
Why do they only divide a few times in tissue culture? Because don't they divide more than that in our body?
Yeah, it's a good question. The cycle in our body is regulated in terms of when they divide, how often they will divide, and so forth. In a petri dish, that's not regulated, and so they go through a lot of generations pretty quickly. Yeah. Yes, Robert. Forgot your question. Okay, I can't answer that one. So, all right. So there's some really interesting things about this. So you might start thinking, well, if the length of a telomere does influence life expectancy, and there are some suggestions that it does, we have to think about this when we think about, for example, what about cloned animals? A cloned animal is made by taking the nucleus of an existing animal, injecting it into a fertilized egg of another organism, removing the fertilized egg's nucleus. Haven't we just put some shortened telomeres into that fertilized egg? Won't that clone have a shorter lifespan? And the answer is that in some cases it appears that this is in fact the case. Okay? In some cases it appears, for example, Dolly the sheep, that was the first higher organism that was cloned, died relatively early of symptoms that appeared to be like old age. Okay? However, there are other organisms and there are other studies that show, in fact, that cloned organisms don't have a shorter lifespan. So the answer, the jury is still out on whether or not this is the case. It's also been shown subsequently in the past year or so that if you look at cloned organisms or you look at the embryo that's developing, they actually do turn on telomerase. So that may help to reduce the effects of the problems associated with shortened telomeres with the original organism. Yes, Jen?
So if most of the cells do not have active telomerase, what cells do?
What cells do have active telomerase? Stem cells. Okay. Stem cells are the main cells that we have that have active telomerase. Okay. There are minor other ones, but stem cells are the main ones that we think about. Yes. Yes, Shannon. I can't pull your name out of my head.
So we talked before about cancer mutations that cause cancer and developing rapidly. Do those type of cancers have to also have telomerase?
Good question. So do cancers have to have an active telomerase? For the most part, it appears most cancers have an active telomerase. There are a few that do not, and they may have some unusual mechanisms of maintaining their telomeres.
So you have to have 2 mutations in order to do that, or just...
So for a cell to become cancerous, that is for a cell to develop a tumor, there is more than one event that has to happen, and that's been known for a long time. Okay? But multiple things have to happen that ultimately results in a tumor. Yeah. For a cell to divide uncontrollably or for a cell to lose its controls, yes, indeed, it's possible that that could be a single mutation that could favor that. However, in order to develop a stable tumor, there may be other things that are necessary. Other questions? Gesundheit. This is cold season. I've had so many people in my office. I'll probably get the cold again. I've had so many people in my office that have been sick over the past week or so. However, I have an immune system of steel, so. Famous last words, right? If I come in on Wednesday and I'm hacking and coughing, you guys can laugh at me, okay? So, yes, Grant.
What facilitates the telomerase to bind?
What facilitates the telomerase binding? That's a good question. I believe it's simply a free 3' end. I don't know the answer to that question. That's a very good question. Okay, so that's what's up with eukaryotic cells. Telomerase is a very interesting and important enzyme. And oh, by the way, I haven't told you the flip side of that. You might say, well, okay, if telomerase is active in cancer cells, can we kill cancer cells by inhibiting telomerase? That's the other way of looking at it. Instead of turning on telomerase in all cells, what if we knock out telomerase in cancer cells? Can we kill cancer cells? And there are some very promising drugs on the market that appear to do just that. So while they don't stop all cancers, they do appear to be very effective against some types of cancer. And I think you're going to see some of those make it to market. They're in trials right now. Okay, well, let's turn our attention then from replication to some problems that arise in replication. Okay? What you see on the screen is an example of a problem in the process of happening. Okay? This is a problem in the process of happening. How did this guy happen in the first place? Well, first of all, let's look at the sequence that we're studying. This sequence, we have various regions of DNA in our cells that have what are called repeated regions. They're not telomeres, they're shorter. These guys are about 3 base pairs long. Some of them are in important proteins. They're actually there for a reason. Okay? Well, if we have a repeated region like we see here, CAG, 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 blah, 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 it turns out that this GCA could pair with this CGT, or if it slipped, it could slide back and pair with this one. And in fact, what's happened is this top strand has, in fact, slipped. When it slipped, everything else down here ended up being base paired properly. Well, if replication was going on while that slip occurred, the top strand, when its replication is done, will be longer than the bottom strand. Okay? Because it slipped backwards and then just this guy got replicated, got extended a couple times more than it should have. Okay? This type of triplet repeat, okay, is a target for DNA repair enzymes. Okay? In people who do not have Huntington's disease, they are able, our DNA repair system is able to recognize when such slips occur and fix them. Okay? If you have Huntington's disease, the problem is that your DNA repair proteins do not recognize this and do not fix this. Now, since this is a region of coding for a protein, what we've just done in that protein is we've now put some additional copies of that same amino acid into that protein. All right? We've got a different kind of a mutation. Well, you guys hopefully learned last term when we think about protein structure, if we put even a single amino acid in, we can change the shape of a protein, and when we change the shape of a protein, we can change the function of a protein. Okay? Huntington's disease is a very, very nasty problem. It tends to strike people in their midlife, oftentimes after they have had children. Okay? And it is always fatal. It is very debilitating. It causes some severe impairments with respect to development, physical development, and also some neurological problems that arise from it. The famous singer Woody Guthrie, a folk singer Woody Guthrie, died of Huntington's disease back in the 40s, I want to say. Okay, not before he had a son, Arlo Guthrie. And Arlo Guthrie was also a folk singer. Arlo Guthrie was very popular during the 60s, sang a song called Alice's Restaurant. Anybody ever heard Alice's Restaurant? You can get anything that you want at Alice's Restaurant. That's Woody. That's Arlo Guthrie, Woody's son. Woody had the disease, and the disease is a dominant disease, and as a result, that meant that Woody had one good copy and one bad copy of the gene. That meant that Arlo had a 50-50 chance of contracting the disease. In the 60s, they had no way of analyzing the DNA and knowing whether or not Arlo was going to succumb to the disease or not. He did not. He got the 50-50 chance, the dice rolled the right way, and Arlo Guthrie is still alive today. So Huntington's disease arises from problems as I show you here. One of the reasons it persists in the population is because it usually strikes, its effects come after reproductive years. Okay. Other DNA damage, that slipping is one that's sort of built into our own cells, but there's chemical damage that can occur to DNA. The chemical damage that can occur to DNA, one example you see on the screen, is an oxidation of a guanine base and creates something called 8-oxoguanine. 8-oxoguanine is very much like guanine, except for it's possible to pair 8-oxoguanine with adenine. Now, that means that when DNA replication is occurring, if the cell has 8-oxoguanine on one strand, when the DNA polymerase comes along, it has a reasonable chance, and I say reasonable chance because it can still put C across it, but it has a reasonable chance it will put A across from 8-oxoguanine. Well, if that happens, of course, we've just had a mutation. What should have had a C now has an A in its place. When this strand goes and when this other strand goes and replicates, it's going to have an A. It's going to now make for a T. So we've had a mutation that's happened as a result of this. Our cells and the cells of bacteria have ways of recognizing and removing 8-oxoguanine. Okay? They have ways of removing 8-oxoguanine. I'm going to show you a bit of that in just a bit. Okay? All right. Other things that can happen. Deamination, losing an amine group off of adenine gives hypoxanthine. Okay? When this happens, the cell has to do something with this altered base that's in the DNA. So there are situations I'm showing you where we have all chemical alterations to the DNA, and those guys have to be fixed. Here's another thing that can happen. Aflatoxin. Aflatoxin is a toxic substance that is produced by some molds, and the most common molds that produce it infect peanut plants. Okay? If you eat a lot of peanuts, you may be much more susceptible to aflatoxin. Aflatoxin is one of those compounds that is a DNA adduct. This guy will react with bases in DNA, cause a big bulge in the DNA, and favor mutation. Aflatoxin is, as you might expect, a carcinogen, and because of the mutations that it favors. Thymine dimers, okay? Thymine dimers arise from ultraviolet radiation hitting two adjacent thymines in the same strand. Two T's on the same strand. So I have a T next to a T on the same strand, and I hit it with ultraviolet radiation, a covalent bond between these two can be formed. Well, this covalent bond is not natural in DNA. The DNA polymerase, when it's going to replicate this DNA, stops when it hits that covalent bond. It doesn't know what to put across from it because it's not seen something like that before. Okay? How do we get thymine dimers? Ultraviolet light. Too much sun, too much tanning booth. Okay? If you're young, one of the dumbest things you can do is go climb in a tanning booth. Absolutely. Okay? I like to compare it to taking up smoking at your age. It's not a good idea. You're favoring a lot of mutation. Shannon?
Why are they mirror image of each other?
That is just the way they've drawn it. They've just drawn it. So they're actually sitting one on top of the other. They're trying to show that this part hits, and it's just sitting above it like this. They can't draw it in 3D. Yeah. But they would actually be sitting on the same strand just like that. Okay. So tanning booth exposure, too much sunburning, etc., causes real problems. Be careful with that. Last, there are cross-linking agents that can react at both ends. So you can have one bind to one base on one strand, the other side of it bind to another base on another strand, and now make a covalent bond between the strands. Those strands can't be peeled apart. All right? This guy is a common component of tansy ragwort, which is a nasty weed that livestock sometimes eat. Is there a question over here?
Yeah, I was wondering with thymine dimers.
Yes.
If you live in a really sunny area, and if you put on sunscreen, will that help you protect from? Will that protect you from thymine dimers?
Will sunscreen protect you from thymine dimers? Sunscreen will reduce the level of UV exposure, and the answer is yes, it will. And being out in the sun a lot, putting on some sunscreen is a good idea in that respect, definitely. Okay, well, those are some of the types of chemical damage that can occur. Let's look at the DNA repair systems for fixing them. Okay? Well, the first repair system that we talk about has nothing to do with those things that I told you about there. That is, this repair system doesn't fix problems with DNA damage. Instead, this repair system is one you've already seen. It's proofreading. When I described proofreading, you may remember I said it was an activity that was known as the 3' to 5' exonuclease activity of DNA polymerases. And this activity removes errors as they are being made in the DNA. So if the polymerase, for instance, accidentally puts a G across from a G, the proofreading activity will back it up, okay, and then come back and replace it with a C so that everything is okay. Now, proofreading is not perfect. Just like your own proofreading of your papers is not perfect, so too is proofreading not perfect, but it's pretty darn good. As I said last time, it improves the fidelity of the replication by about 100 to 1,000-fold. It does a very good job of helping to ensure that this thing has been copied as accurately as possible. This figure is, I think, not a very good one, but I'll tell you, it does illustrate an important principle. When the polymerase is moving along and an error occurs, okay, the polymerase actually has to back up a fair stretch to get that base into the exonuclease site. You can see several bases had to be peeled away. So it's for that reason I said when the polymerase is moving along, replicating, and it said error, I said it had to back up. The reason it had to back up was it had to peel enough of that strand off so that the bad base is inserted into the exonuclease activity of the enzyme. Okay. Well, once that happens, of course, that bad base is removed, and then the polymerase can go back along its merry way and replace things. Yes, Jen?
So if it's a few base pairs down, how does it, is there some lagging part of the protein which says, oh, hey, we have to stay here?
It's a very good question. How does the protein know that there's a problem there? And the protein knows by virtue of the fact that the base that's there bulges. And so the protein, when it gets that bulge, actually has a signal that there's a problem. It's a very good question. And that's been very, very intensely studied for that very reason, because they didn't know originally that this thing was further away. They assumed it was right at the same place, and it's not. Okay, well, that's DNA proofreading. Mismatch repair. Let's talk about things now that escape proofreading. Let's imagine I've gone through I have replicated my DNA and I made an error, and that error escaped proofreading. It does happen, just like your own proofreading of your papers doesn't catch every mistake that you make. Well, when you've done that, what happens is that this error is now embedded in the DNA, sort of like this GT mismatch that you see right here. Okay? The system I'm going to describe to you is again from E. coli because it's the most studied. This DNA mismatch repair system, and that's the name of it, by the way, mismatch repair system. This mismatch repair system uses 3 proteins to remove that problem. The first of these is a protein called MUT-S, M-U-T-S. MUT-S is a protein that looks for bulges. It finds that bulge in the DNA and says, oh, there's a mismatch, there's a problem here. So MUT-S binds to bulges. MUT-L is a protein that binds to MUT-S, and its primary function is to provide a docking place for the last protein, MUT-H. MUT-H is an endonuclease, okay? And it cuts a strand, and then it's both an endo and an exo. Its exonuclease removes that stretch. So we first have to make an endonucleolytic cut, that is, we have to break a phosphodiester bond, and then we have to excise backwards. That leaves a gap like you see here, and the DNA polymerase comes along and fills it back in. Notice it's polymerase 3 that actually fills it in. As a result of that, the damage is repaired. Now, one question that arises from this is, well, how does the cell know which strand had the error? It's equally likely it could be in the G or it could be in the T. And if I flip a coin, I'm not doing any good, right? 50-50 chance, a 50-50 chance of it coming out okay when I replicate here anyway. How do I know, how does the cell know which one it does? It turns out E. coli cells have a very interesting system for recognizing what is the newest synthesized strand compared to the original parental strand. Okay? So if the cell knows which one is the new strand and which one is the old strand, then the cell knows which strand to cut and remove pieces from. Are you with me? So, the E. coli, first of all, I will tell you, has a system for identifying which strand is new and which strand is old. Okay? That system relies upon, and I don't have it there, sorry. It relies upon a sequence that is methylated. Okay? What does that mean? Well, it turns out that all cells have enzymes that will put methyl groups onto DNA. All cells have enzymes that will put methyl groups onto DNA. Okay? The sequence that E. coli puts methyl groups onto is GATC. Okay? GATC. Now, that is a symmetrical sequence. If I go GATC this way, it pairs with GATC this way. Right? After I have replicated my DNA, the methylation system comes along and it starts methylating everything. Okay? Before the methylation system comes along, the top strand will have methyl groups on its GATCs. The bottom strand will not, because it's been newly replicated and the methyl has not been added yet. So what this guy has to do is it has to bind to the strand that has the methyls in the GATC. Now, GATC on average occurs about once every 256 base pairs. That's not very far. So as long as we're within about 256 base pairs of the problem, the cell knows where to bind, what to do, and how to remove the stuff. Yes, Stephanie?
Didn't we also hear about a similar process for bacterial immune system sort of response?
Oh, a very good question. So her question was, she's remembering from last term, I talked about methylases relative to restriction enzymes. Mm-hmm. So in fact, that's exactly right. Cells use methylases to protect their own DNA against cutting their own DNA. So it's another place where methyl groups are useful for bacterial cells. That is, however, different from this in the sense that in that case, it's protecting against cutting of its own DNA. In this case, it's actually telling the replication system which strand had the error. Okay? Good question, though. Yes?
So the has to recognize the bulge and find the right strand.
That's right.
Does that mean that it's 256 base pairs big at least?
Oh, good question. So does that mean that it's at least 256 base pairs big? No, it's not. But it means it has to have influence over at least about 256 base pairs of DNA. And it does. So this DNA, it turns out, can curl, it can make circles and so forth. And so there's all kinds of ways of getting sequences that aren't very close to each other closer by circling them around or something. But a very good question, yeah. This turns out to be a very big issue when we talk later about eukaryotic transcriptional control. Because in some cases, eukaryotes are able to control genes from thousands and thousands of bases away, even though the proteins themselves are much smaller than that. Yes, sir?
Most of these examples, you start with E. coli.
Yes.
But is this most everything you're saying true for eukaryotes?
Good question. So is most of what I'm saying here true in eukaryotes? The answer is yes, they are, and they have different names. But and the system that we use in our cells is very, very similar to this. In fact, we call ours mut S alpha, for example. And it's a little bit more complex in terms of the number of proteins that's involved, but the same basic principle holds. But yes, they are very, very similar in us. Good question. Okay, I've got just a couple minutes. Let me get to one more thing, and then we'll finish this material on Wednesday. Okay. Let's imagine I've got a DNA adduct, one of those chemicals that I put onto DNA. Let's imagine, alternatively, that I have a thymine dimer. I need to get those things out of there. Okay? In either case, I'm going to have, again, a bulge in the DNA, but this bulge is going to be rather different than the mismatch bulge. In some cases, the bulge is going to be very large. And when that happens, a different system is invoked. The different system here is called nucleotide excision repair. Different set of proteins doing different things. Okay? In this case, we don't care which was the parental strand. We know where the problem is because the problem is a chemically altered base. That chemically altered base, we have to assume, is where the problem resides. That chemically altered base is removed by a system called an exonuclease, E-X-C-I-N-U-C-L-A-S-E. It basically does what you saw in the last system. That is, it cuts on a strand, it excises on that strand, and then this guy gets repaired by DNA polymerase, ultimately rejoined by DNA ligase. The main difference here is the recognition. Okay? We don't have to decide which strand has the error. We know where the error is. We get removal, and then we get repair of everything here. Okay. Yeah, one question, yeah.
Why is it DNA polymerase 1 and not 3?
Why is it 1 and not 3? Just the way the system evolved. Yeah, that's a common question. I would actually predict that the other one